So here we see the enzyme with an open active site. The substrate, represented by red Play-Doh, binds to the enzyme's active site. A new product is then formed after the red substrate binded to the enzyme's active site, and this new product is represented by the green ball of Play-Doh.
In the first picture shown, we see a lone enzyme with an open active site. In the second picture, a substrate and a competitive inhibitor are approaching the enzyme's active site, both with the intent to bond with it. In the third picture, the competitive inhibitor binds to the active site, which makes it so the substrate can't bind to that enzyme anymore. The fourth picture shows how the substrate cannot bind to the enzyme since the competitive inhibitor is there. The fifth picture shows the substrate moving along in efforts to find another enzyme to bond with. This process is called competitive inhibition. It is called such because the enzyme inhibitor in this process competes with the substrate. In the first picture, we see an enzyme with an open active site and an open non-competitive inhibitor site. The second picture shows the non-competitive inhibitor bounded to its site. The third picture then shows the active site closing due to the non-competitive inhibitor being bonded to the enzyme. The fourth picture shows that the substrate now cannot bind to the enzyme to become a product. The fifth picture shows the substrate leaving the closed enzyme. This process is called a non-competitive inhibition. It is called non-competitive inhibition because the enzyme inhibitor in this process binds to a site other than the active site, with no need of competing with the substrate to bind to the active site, in order to exchange the shape of the active site. In the first picture, we see the enzyme's active site partially closed, blocking substrates from binding to the activation site of the enzyme in order to become a product. In the second picture, the allosteric activator comes close to the enzyme's allosteric site. The third picture shows the allosteric activator bonded to the enzyme's allosteric site. The fourth picture shows the allosteric activator causing the enzyme's active site to open, allowing substrates to bind to it. The fifth picture shows a substrate approaching the enzyme's newly opened active site. The sixth picture shows the substrate binding to the enzyme. This process is called allosteric regulation. In the first picture, we see an enzyme with an open active site and an open inhibitor site. The second picture shows a substrate 1, bonding to an even active site. The third picture shows the new product 1, leaving the enzyme, with the new product represented by a blue piece of Play-Doh attached to the top of the red piece of Play-Doh. In the fourth picture, a new substrate 2, the previous picture's product, approaches the enzyme's open active site. In the fifth picture, substrate 2 bonds to the enzyme's active site. The sixth picture shows product 2, which was created from the bonding of substrate 2 to the enzyme's active site, leaving the enzyme. In the seventh picture, product 2 comes back to the enzyme's inhibition site and bonds with it. The eighth picture shows product 2 acting as an inhibitor by changing the shape of the enzyme's active site to be enclosed. The ninth picture shows a substrate 1, approaching the enzyme's closed active site, with the inhibition site filled by product 2 acting as an inhibitor. In the 10th picture, a substrate 1 is shown as not being able to access the enzyme's active site. The 11th picture shows substrate 1 leaving the enzyme because it cannot bond with it. This long process is called feedback inhibition, in which the regulation of an enzyme occurs by having the final product of an enzyme acting as that enzyme's own inhibitor.